Thank you very much. So my interest is tropical immunology, aiming to integrate Oxford-style research in the lab with what's already going on in the tropics and finding ways to slot in with existing large studies in a practical way. And immunology can offer a lot to tropical medicine, most obviously diagnostic tests, which are often based on antibodies, vaccine design, but also understanding disease process as well, looking for new drugs, immunotherapeutics, understanding the environmental niche of bacteria and other pathogens, and can also play a role in understanding antibiotic resistance. My research focuses vaccine development specifically for intracellular pathogens. I did my PhD just nearby here with Adrian Hill on malaria vaccines, where I did clinical trials because I'm a medic as well, and I did immune correlates for protection against malaria vaccines, in particular gene expression profiling. But I wanted to go out to Thailand, and I decided to focus on some of the neglected tropical diseases out in Thailand. So in particular, characterising the immune response to melioidosis, which I'm going to talk to you about today. I'm also, because of melioidosis and its interaction with diabetes, which I'll tell you about, that has got me very interested in diabetics and what it is about people with diabetes that make them more susceptible to some diseases more than other diseases. So I spent 4 wonderful years out in Bangkok. Has anyone been out there? Yeah, it's really, really amazing place. There is a big research unit in Bangkok itself, which hasn't got direct clinical care there, so it's a hub. And most of the researchers there, a mixture of Thai and international staff, I think something like 30 nationalities last time we counted, come and go around the region. A lot of collaboration with the Vietnam unit and the African units as well. A lot of work in the Thai-Burmese border by Francois Noston, and in Cambodia, now led by Paul Turner, and up in Laos with Paul Newton. I have to say the main thrust of the Bangkok unit is malaria. It's very much in the heart and soul of the unit. Wonderful body of research built up by Nick White. And if you're not working on malaria, you can feel a bit left out. So I ended up doing a little bit of malaria immunology on the sly, even though it wasn't really what I was there to do. And a lot of the other departments at Moru are actually quite geared around malaria, in particular pharmacology, the mathematical modelling, and the excellent trial support unit. But they're increasingly interested in TB, antimicrobial resistance, emerging pathogens. And the microbiology department, which is now led by the incredibly able Direk, and was really developed quite a lot by Sharon Peacock before him, spends a lot of its energy on Melioid, but also looks at scrub typhus and Staph aureus lepto and resistant bacteria. I spent a lot of time in the northeast of Thailand, up in Ubon, Ratchathani. This is rice farming country. It's one of the most rural parts of Thailand. It's one of the poorest as well. Nearly everybody is connected to rice farming. People they essentially live on rice farms. And if you visit someone there, you often have to park your car and then traipse through a rice field to get to their door. So most of the population, apart from the very urban people around the town centre, have exposure to rice fields. It's an upper middle income country. So the hospitals there are pretty good. If you're used to spending time in African hospitals, which I had before, it's a different world. They have ITUs, they have a lot of equipment, they have a CT scanner, they have an MRI scanner, but not everybody can access everything. It is a resource limited setup, but they work pretty efficiently with what they've got. So in Thailand, I have a small group in Bangkok, and a nice laboratory there, including flow cytometry. And then I set up a little satellite lab up north in Ubon, Ratchathani. It's a very little lab anyway that is a Melioid lab looking at the bug. So I had a little corner which I tried to make sterile for immunology. I've now since July moved back to Oxford. I'm very happy at Medawar, surrounded by Excellent immunologists led by Paul Klenneman working on Hep C, Hep B, John Freitas there working on HIV, so that's really useful. And wonderfully, some of you guys are moving down there, so there'll be more tropical medicines, so I'm finding that a nice home. I'm very thrilled to have new staff, so I have Barbara here, and she's a postdoc immunologist. Very lucky to have Barbara, who's helping me a lot set up the lab. I'm also very lucky to have Rabi here, who is a doctor from Bangladesh with quite a lot of experience in clinical trials, but he's come to Oxford to learn immunology on a Commonwealth scholarship. I also have a Thai student called Palm, who's currently on a Wellcome master's studentship at the London School, learning immunology, and he'll come to Oxford for a few months before returning with his new skills to Bangkok. So melioidosis is a nasty, nasty disease. It's a gram-negative sepsis. The bacteria is related to Pseudomonas. It's an environmental bacteria that lives in the soil. It's a major killer in northeast Thailand. And the mortality in northeast Thailand in that hospital is about 40%. And this is patients who come to hospital and are culture positive, so get it diagnosed, which is an appalling mortality. And that's in spite of the hospital being geared up to it and putting everyone on the appropriate antibiotics. It's an emerging pathogen elsewhere, as I'm going to explain. And there's actually quite a defined at-risk population. So in Ubon, 2/3 of the patients there have diabetes. And the rest have alcohol excess, or they're elderly or immunocompromised in some ways. But it's interesting, I can actually tell the patients apart on the ward. The diabetics with Melioid tend to look healthy. They're healthy people who happen to have diabetes. They've got this chink in their immune armour. And the non-diabetics tend to look like frail people with medical problems, either because they're alcoholics or because they've got cancer or something else. So they're quite different populations, actually. It's also a major problem in northern Australia, so around Townsville, particularly in the Aborigine community. And the clinical course is nasty. I've seen many, many patients. This was one patient in particular who went to ITU. He went on meropenem, the IV antibiotic, which is appropriate. But you can see this is his temperature chart. And he just carried on spiking fevers for a week. And then after a week, he was still spiking fevers. If you scan these people, you can't find a big load of pus to drain. So usually as an infectious diseases doctor, if you see that, you think you're missing a big abscess or something. They have micro abscesses in their liver and spleen, but nothing that you can drain out. You've just got to give the supportive care and keep treating with the antibiotics till they pull through. And this particular patient was really sad because he was from Laos and he was an immigrant worker, and after 10 days his family took him home. They just said he's not doing well. And I don't know if in this case, if we'd stuck with it, we would have won. Some people can pull through after this, but the mortality is terrible, and in a resource limited setting as well. So a vaccine is highly desirable. As I said, it's caused by Burkholderia pseudomallei. You get it from inhalation, so particularly during the rainy season, it lives in the soil and the rain whips it up. You can get it in your lungs. After the Vietnam War, some of the veterans back in America got Melioids. Sometimes years later, the record is not from the Vietnam War, but there's a case described 62 years after somebody left a region, so it can be latent. And the Vietnam vets, it was particularly the helicopter crew, so presumably the helicopter stirred it up. So inhalation is the major route, but it's actually been shown to also be spread by inoculation through cuts in your feet and through drinking contaminated drinking water. Usually it's people immunocompromised, but anyone can get it if they get a big enough bioburden. Kids get it, and they get a different disease. They get a primary disease, their first infection, and they probably get it from playing in the fields and going, kids going face down in the mud and really getting a mouthful of it. And they get parotitis, the glands here, the ones that go up in mumps, get very swollen. But they usually have a less severe illness. It's typically not fatal in kids. You can also get it in healthy immunocompetent adults. In certain circumstances, Sharon Peacock wrote up a case series after the tsunami of tsunami survivors, presumably again got a really big bioburden. It's been described in triathletes in the region. So it can happen, and it's the classic triad, whether you get disease of the virulence of the bug, your immune status, and the dose. Melioidosis in northeast Thailand. This is a nice paper from Direk, that's his surname down there, Limmathurotsakulet. He wrote this in 2010, and this was looking at the mortality rates from infections in Ubon. And you can see, I haven't got a pointer, but Melioid is the black line in the middle. You can see the biggest killer then was AIDS. So this is in pre-2010. AIDS deaths have come down somewhat as more antiretrovirals are used. And then it was up there with TB as a killer. Estimated incidence about 20 per 100,000, but if you look specifically in diabetics, it's between 150 and 250 per 100,000. Mortality in that hospital, in that study, was 42%, and sadly that hasn't come down since 6 years later. And that hospital has 400 culture-confirmed cases every year, and probably double that number where we don't manage to grow the bug. What's frustrating is, so 400 culture-confirmed cases from one hospital, about over 200 deaths, and we faithfully report them all to the Ministry of Health in Bangkok. Ministry of Health website for all of Thailand, they say the number of deaths from Melioid for the whole kingdom is 10. And we have over 200 from this one hospital. So Direk, who's great at the politics, went to see them and said, why do you do this? Why do you say this? And you know, it holds us back getting funding and demonstrating the problem. And they said, please find us a solution, and then we will publicise the problem. But we can't publicise the problem until we've got the solution. And you know, it's partly concerns about their food industry and tourism and things like this. So this is what we're up against. I'm not an epidemiologist, and I had some funding interview, and the night before you get desperate measures. I sat there with Wikipedia and added up the population of the world where people have had Melioid, and it's huge because you get it in very populous countries like India. So it's possible that over 2 billion people are exposed. Actually, this is nonsense. Nick White says if you go to an infectious diseases conference and listen to how many of us are exposed and how many of us die from each bug, we'd all be dead several times over. So I know this isn't right, but I'm just trying to say it's a widespread problem. Fortunately, there's better epidemiologists than me. So this is Direk, my head of department in Bangkok, who is such a clever and hardworking person, and he has done this amazing study in collaboration with Simon Hayes, who used to be here, and kind of like the famous Bob Snow paper on malaria, and it's also been done for dengue, trying to model what is the real global burden. And you can say that all models are wrong, but some are useful. And where there is no data, it's a very useful starting point for a conversation. So this is a really beautiful study. It was a lot of work. And what he did was he looked at where Melioid had been described clinically and in animals. It's a major veterinary pathogen as well. And he looked at the soil in those areas, taking into account things like the pH and the humidity and the mineral content and the rainfall and all these things. And then built a model using, there's actually a lot of data worldwide on soil. It must be to do with agriculture, but there's a lot of data on soil types around the globe. So he was able to predict on soil type and also bringing in information on where the Melioid had been seen before. Countries which he reckons have it, where he is now organising to send research groups to do soil sampling and increase surveillance in the hospitals. And he also used this to estimate mortality, which is quite a complex thing to do, and he based it on childhood mortality rates. And he estimated there to be about 165,000 cases worldwide, and the worldwide mortality to be 89,000. Now you see there's wide confidence intervals there because it's a model. But that's actually an incredible amount. It's higher than dengue. It's up there with measles. And it just shows this is potentially a very neglected, very global problem. It also shows what a problem it's likely to be in India. So that's a real focus for research. You'll also see Bangladesh is pretty hot as well, which is why I'm thrilled to have a doctor from Bangladesh to work on this. And Nigeria, a potential hotspot as well. So it's a good starting point. This paper came out in Nature Microbiology last month, and happily there was lots and lots of exposure for it. It made the Wall Street Journal. It's up there with Simon Cowell as newsworthy. The BBC World Service made a podcast you can listen to. It's in The Scientist, and there was an editorial in Nature on it. So that's all good for Melioid. So it's a neglected tropical disease. WHO has 17 priority neglected diseases, and this is not on the list. But there is increasing recognition. We believe, unfortunately, the incidence is likely to rise, partly because of changing land use and the rise in diabetes in affected regions. And also there's a big change globally in the people who do the farming. So in Thailand, China, the rice farming is now increasingly done by an aging population as the youngsters urbanize and move to urban areas. And we reckon a vaccine would be cost effective. This is cost effectiveness modelling done by Joel Lubel, our health economist at Moru. And essentially it depends on the incidence, but if you targeted diabetics, where the incidence is felt to be 250 per 100,000 in Northern Australia, for example, if you had a vaccine that lasted 3 years and was 50% efficacious, it would be cost effective, depending on the price. And if you had a vaccine that lasted 10 years, then it's easily cost effective. So a vaccine for Melioid, the key questions are, for me, as someone with a special interest coming from the Hill Group background in cellular immunology, is what is the role of cellular immunology here? A lot of research into Melioid immunology before me was very much on the easily measurable bits, which happen to be the innate side. So there's quite a lot of research on toll-like receptors and things like that, but you're not going to get a vaccine by focusing only on the innate side. You need to look at how your immune system can learn. And it's a bacteria that hides inside cells, so I believe you need your T-cell immunity to fight that. And I wanted to see how T-cell immunity related to mortality and to diabetes. And I wanted to zone in on the antigens to put in a vaccine and try to understand what it is about diabetics in particular. And can we learn something for other diseases as well? So I first started thinking about this in 2008, and I moved out to Bangkok in 2011, and the goal was to set up the capacity and establish the longitudinal clinical studies and start looking at the immunity. So the study I set up started in 2012. It was a longitudinal study in the northeast. We have 200 cases of acute melioidosis to date followed up, and a lot of control subjects as well. But we've got another study starting with an aim of reaching 600 in total. So it was difficult to set up there. As you can imagine, many people in this room have a lot of tropical experience. I know Catherine is particularly talented at working in these, trying to get things going. And it was difficult, but some things are easier, and you get happy surprises. So it's difficult to move samples around. You know, it's difficult to move samples around in the UK. It's really easy in Thailand. So it's an overnight bus. In Bangkok, we would buy a box of dry ice and put it on the overnight bus for £2. And there's lots of people around to help, so somebody would meet it off the bus and take it to the lab, and then we'd put our samples in it and then put it back on the overnight bus for another £2, and then it would come back to Bangkok. And we did that every 2 weeks. You know, we've been doing that for 3 years. We never had a problem. So some things are easy. The protocol needed ice, and I was like, we don't have an ice machine, and everyone was like, strange thing to worry about, because they just go outside onto the street and from the drink cellar in the foyer of the hospital buy a bag of ice every time you need ice. So not everything's harder, but we managed to get LE spots going there, eventually good quality. Setting up the Bangkok lab was hard as well. So on the left, I wanted to get flow cytometry going, and on the left is my flow cytometer, which sat in the corridor of the institute for 3 months because we had a military coup, and it could only be installed by these engineers from Singapore, and they wouldn't come because they felt it was dangerous. And the worst thing was that everyone was pestering me about it, going, oh, it's getting in the way, you know, when are you going to do something about it? It's like, I know. But we eventually got it set up. And flow cytometry is difficult because it takes a long time to order everything in Thailand. We get it all shipped by John Minogue here, who's wonderful, and he ships our supplies out, but the sort of ordering to in your hand time was about 3 months. And when you're trying to design antibody panels, it's actually quite trial and error. They're £300 a vial. This was a 10-colour fax machine, so you want 10 different things to match, and you try one, it doesn't suit you, and want to try another. So we wasted a lot of time and money sorting that out. But we managed to get there, and it was fun and rewarding, and my Thai team are very smart and hard-working, very keen to learn. So I've got 200 patients that I've tracked, and this is where the Melioid was identified from. It was half of them had it in the bloodstream. Out of the 200, 32%, a third died, which is just terrible. Also bearing in mind that these patients are recruited on average 4 days after they come to hospital, because I don't recruit them until I've got the positive culture result. So it's an appalling mortality. 2/3 of them had diabetes, typically in their 50s. 4 out of the cohort of 200 have relapsed to date. The quoted relapse rate in the literature is about 10 to 15%, but the hospital is following everyone up tighter, trying to get them to take 3 months of oral antibiotics, and so the relapse rate fortunately has gone down. And you can see that some of the deaths, the red line there is 28 days, and I tend to use 28-day mortality as my outcome measure, because some of these people are very frail and they may be dying of other things, but you can see that there's some late deaths. What I found was that the people with known diabetes who come to the hospital have terrible control. So this is HbA1c, which many of you will be familiar with. It's a measure of how sugared your hemoglobin in your blood is. And if you don't have diabetes, your HbA1c is about 6% or below. And if you've got diabetes in the UK, you aim to have an HbA1c of below 7. And people who, the third bar along is people we know have diabetes when they came to hospital, and they had an HbA1c of over 10, which is appalling control. And one of my control groups was diabetic outpatient control, who actually had quite well-controlled diabetes, so it is possible up there. So these are people with poorly controlled or undiagnosed diabetes. And I've actually wondered, and this will be very difficult to explore, but there's actually some interest in the interaction between diabetes and bugs, particularly in TB. There's an interaction there. And there's now literature showing that if you've got TB and you've already got diabetes, your diabetic control gets worse. And if you're diabetic, you're more likely to have TB. And I'm just wondering if there's a lot of Melioid about it. It might actually be contributing to the diabetes burden as well, if a lot of people are infected. People make antibodies when they get melioidosis, but it doesn't seem to help. And I found T-cell responses, and I think only Sarah will appreciate the next few slides, so I might go through them quite quickly to avoid everyone else going to sleep. But essentially, quite strong T-cell responses to killed bacteria were measured, and you can still see them at 1 year. And these responses were, the interferon gamma that I measured was from a variety of cells, but particularly CD8. And I found an association with survival. So the people who died tended to have lower responses than people who lived. And when I show this at meetings, all the mouse immunologists who work on perfect little models say, but they overlap. And I say that's because they're people and they have other things going on as well. But I was quite pleased to see this relationship, and to me this is evidence that your T cells are important in controlling the disease. And again, both CD4 and CD8 cells seem to be important, particularly CD8, and actually nobody who died made a decent CD8 response to this bacteria. Direk helped me do a logistic regression to try and put this data in the context, and what I found was that the LE spot response is important in predicting survival, alongside age, diabetes, renal disease, and new neutrophil count. And also found that people with diabetes had a very sluggish response to Melioid when they were ill on this T-cell assay, which was quite interesting, and that if they survive Melioid, they then have responses like non-diabetics. So it seems to be a problem for diabetics getting a rapid enough T cell response. A higher neutrophil, if you have a lot of neutrophils going on at the time of your infection and then you survive, it's associated with lower long-term cellular responses. So this suggests that the environment, the immune environment, when you get disease, determines your long-term outcomes in terms of your immune response. So the summary of that study was that I saw cellular responses during acute disease which was still detectable 1 year later, and that high responses were associated with survival. People with diabetes have lower responses, and high neutrophils during acute illness are associated with a lower long-term cellular response. So I believe that the T cell responses are important in defence and contribute to susceptibility in diabetics. And we've published that one now. The next project was a big project trying to zone in on which proteins to put in a vaccine. And this was a collaboration with Rosemary Boyton and Danny Altman at Imperial College. We looked at 15 proteins from the bacteria, trying to decide which one or several to put in a vaccine. And a huge amount of work was done, but essentially 5 proteins were particularly associated with survival. And we looked several different ways. We looked at how long the responses to these proteins lasted. And we also looked in London at people with cystic fibrosis. And the reason we did that is because there's a bacteria or a group of bacteria called Burkholderia capacia complex. That's the same genus, so like a cousin. And this bacteria infects people with cystic fibrosis and gives them a worse outcome. So we'd be very keen for a vaccine for Melioid to also work for kids with cystic fibrosis to stop them getting colonised with this devastating bug. And we found that some of the proteins we looked at were also immunogenic in people with cystic fibrosis. And overall, if we sum up the responses we looked at, we found that people who survived melioidosis had around 2,000 spots on average, a very high response to the sum of these 15 proteins. And it was much, much higher than people who died. And we didn't see this with the control panels we looked at. And there's a bit, we're trying to write this up for publication, but there's some further gene expression work underway. I did quite a lot of other things out there, and one thing in particular I've been trying to do is really develop assays that are easy to integrate with other studies. So while all the years I was in Moro, other researchers said, can you do some immunology for me? And if you zone in on what they're after, sometimes they actually just want me to measure antibody responses to childhood vaccines, in which case I would just advise them to send them to a service lab because they would do it better than me. But really, when I started talking about larger volumes and isolating peripheral blood mononuclear cells, people's eyes glaze over and they want you to do something with very small volume and minimal processing. And so that actually became a parallel research interest of mine, is trying to develop assays that are just easy to set up. And so this was a field study I was involved in where we were trying to, it was one of Derek's actually, he had a van going around all these fields, and it would just rock up at a field and sample the soil to see if Melioid was there, and then round up any farmers associated with the field and persuade them to give blood to see if there's any correlation between immune responses of the farmers and the presence of bacteria in the field. And so I thought this was great and I got involved. But it was kind of tricky. We were trying to do a whole blood assay for flow cytometry where you just take a very small volume and you stimulate it in the back of the van, literally. And then you incubate them. There's a portable incubator that plugs into the car, the cigarette lighter in the car. And then you can drive around. And we had to make it 48-hour incubation so that, because they were on the road for 2 days. And that's the time the maximum time they'd be on the road before they came back to base. And when they get back to base, we'd then take it in the lab and centrifuge it and do things to it and put it in the freezer. And then eventually you could do quite high-tech flow cytometry on it. So we've been setting that up, and that's great fun. Actually, my next step is to try it without the incubator because I figure it's kind of hot there anyway, so we'll see. The next steps for a vaccine will be manufacturing candidates, putting it into a vaccine platform, and that's where you need the expertise of the likes of Adrian. And I've got a close relationship with DSTL down at Porton Down, because this is a potential bioterrorism agent. They do a lot of work there, and they've got the category 3 facilities necessary for challenging animals with candidate vaccines. But I'm a human immunologist, so I continue to focus on the humans and trying to get to the bottom of what we need to do. Got ongoing work in Bangkok, so I've still got a team there, and one of the purposes of the Oxford lab is to support the team there, and in particular develop the flow cytometry panels and make things more efficient. But there's also a lot of work they can do there. We've got a new study where we're really focusing on archiving as many samples as we can. There's a wonderful machine here at Medawar called the Zell scanner, which is kind of like a cross between a microscope and a flow cytometer. But the reason I got interested in it is you get a chip and you put your cells on the chip, and it stores every cell in a unique location, and then you can just freeze it, and then you ship your chips. So it's actually ideal for tropical medicine. Very simple, just to add your cells onto the chip, and then they're stored. And then when you get them back to Oxford in this fancy machine, you can stain them, and this is the wonderful bit, you can stain them again and again and again. So you stain them and have a look, and then you can bleach it all off and start again. It's like magic. So these very precious samples you can collect somewhere relatively simple in terms of technology, and ship them to Oxford and have a look. So trying to develop that, doing work on antibodies, innate immune responses, and the cross-reactivity of other species. And now we're in Oxford, we've got a very large archive to work through, and it's an opportunity to address questions about Melioid and other diseases as well, and also to think about fatal bacterial sepsis. So we've got a long list of questions for Barbara to work through, and trying to develop gene expression profiling in this cohort as well. So in conclusion, Melioid is a major cause of death and morbidity in Southeast Asia and Northern Australia, but actually grossly under-recognised worldwide. I think you'll probably be hearing more about it. A typical patient is a rice farmer in middle age with diabetes. There's a well-defined market for a vaccine. Strong T-cell responses do correlate with survival, so I believe we're going to need a T-cell inducing platform for an effective vaccine. Key protein antigens for vaccines have been identified, and we've also got a field site developed now for vaccine testing. So many people to thank, and please give me some questions.